Hello faculty and students. Welcome to the Riboswitch Reporter Research Group. The Riboswitch Reporter Research Group is part of the Molecular Diagnostics stream. Our research group members are Garmani, Daniel, and myself, Mary. Our stream focuses on the research of aptamers and biosensors. Our research educator is Dr. Spirito, and we also collaborate with Dr. Winkler's and Dr. White's lab on campus. Our website is based on Dr. Rice's research, who was a former graduate student in Dr. Winkler's lab. Dr. Rice conducted her research on the effect of cyclic diGMP in gram-positive bacteria. A Riboswitch reporter is a biosensor. So what is a biosensor? A biosensor is a biological tool to detect molecules in any environment. For the Riboswitch in particular, the environment is a gram-positive bacteria, Bacillus subtilis. Most biosensors work in very similar ways. Some of the examples of common biosensors are glucose and pregnancy tests. The difference in each biosensor depends on the molecule being detected, the molecule used to detect the target molecule, and the expression platform. As you can see in the diagram, a sample such as blood or any fluid or solid or anything that has the biological target molecule can be detected using a biosensor, such as seen in the second image. So a biosensor works by detecting the target molecule using a cell antibody or enzyme which attaches to the target molecule. Once it binds to the target molecule, it gives out a heat transfer, maybe light, or an electrochemical expression. And this data that you can see using fluorescence or other types of images or through numbers, for example, for electrochemical expression, can be used to understand the data. Two very important terms to know in our research is Bacillus subtilis and cyclic diGMP. Bacillus subtilis is a gram-positive bacteria used in Dr. Rice's paper to understand the effect of cyclic diGMP on bacterial growth and other processes. Dr. Rice chose Bacillus subtilis because there was a lack of research done to understand the effect of cyclic diGMP in gram-positive bacteria. Cyclic diGMP is an important secondary messenger in bacterial signaling system. Research has shown that cyclic diGMP can control many bacterial processes such as biofilm formation and motility. As you can see in the two pictures, the above one is a picture of a microscopy image of a Bacillus subtilis, and the second image is a formation of a biofilm. So how does a Riboswitch reporter biosensor work? Well, a Riboswitch reporter has a structure called an aptamer domain. The aptamer domain, or the cyclic diGMP, binds to the aptamer domain. If the cyclic diGMP binds to the aptamer domain, it turns on a terminator signal. The terminator, as you can see on the first image, is turned off. It's in a down position, which means that there's no cyclic diGMP present. Therefore, it will continue transcription upstream, and therefore it will fluorescence. However, in the second picture, as you can see, the target molecule binds to the aptamer domain because cyclic diGMP is present. Cyclic diGMP is the target molecule. This, as you can see, turns on the terminator. And once the terminator turns on, it stops the transcription, therefore there's no color expression. This is a really good example of a biosensor. The expression platform used here is color, and as you can see later in the presentation, there are many types of color expressions, but it can be used to understand how cyclic diGMP affects gram-positive bacteria.
Our project is based off of Dr. Weiss's Riboswitch YFP reporter. She created a Riboswitch YFP reporter that has a plasmid vector backbone and a YFP gene, a yellow fluorescent protein gene, which is circled below on the image on the right. She then transformed one Riboswitch YFP reporter into one Bacillus subtilis cell to detect for levels of cyclic di-GMP. We are expanding on this by switching out the YFP gene she used for different fluorogenic aptamers like spinach 2, dimeric broccoli, and mango 3 fluorogenic aptamers.
For our research project, we utilized two main methods, being the Riboswitch constructs and OFTI and microscopy imaging. Together, these methods allow us to make the plasmids we need in order to test for a specific bacteria or target molecule, and then analyze the results. In order to construct a proper Riboswitch, we start off with a pre-made plasmid construct that comes from Dr. Wise's lab. This plasmid construct already targets cyclic di-GMP and is thus our targeting segment. Then, in our lab, we take this plasmid and remove the fluorescent protein, circled in the plasmid on the left, and add our fluorogenic aptamer, which is in the plasmid on the right. This is our reporter segment. Together, these two main segments make up our Riboswitch reporter. Once formed, we replicate our biosensor using PCR and are then able to start inserting it into Bacillus subtilis. Once inserted, our second method takes focus. OFTI and microscopy imaging are the main methods of analyzing and determining the fluorescence intensity within the bacterial cells. An example of this is seen here in these images from Dr. Weiss's lab. On the left are the different microscopy images that have been edited in OFTI to show the bacteria fluoresce and the data we are able to collect from them, comparing fluorescence between different riboswitches. On the right is simply more images displaying different expression platforms on the same bacterial colony and the graphs we are able to collect from them.
Overall, our goal is to create new Riboswitch reporter constructs with the same plasmid vector backbone from Dr. Weiss's Riboswitch reporter. Using different fluorogenic aptamers, we are continuing the search for cyclic di-GMP levels in single Bacillus subtilis cells. We're trying to determine if fluorogenic aptamers are better expression platforms than YFP, and if so, which fluorogenic aptamers. We finished this all off by using microscopy similar to the one Dr. Weiss used in her research. This semester, we created a website for our research project. Our website has 4 main pages: the topics pages, the methods pages, the research page, and pages with additional information. Our topic pages are meant to give you a little more detail about the key topics related to this project, like what cyclic di-GMP is, what riboswitches are, and the difference between fluorogenic aptamers and fluorescent proteins. Our 2 method pages will help you better understand the process of how we build the Riboswitch reporter constructs, as well as the microscopy we perform to obtain our data. Our research page shows Dr. Weiss's research and how we used it, along with future research ideas based on our project. In the additional information section, you can find information about our team, general FAQs about our project, and the resources we used. Thanks for watching, and we hope you enjoy learning about our project through our website.